So from chapter 5 on viruses. So we finished talking about bacteriophages, and now we'll finish this discussion talking about animal viruses. Because I'm doing this voiceover, I will cover culturing techniques, although on the lecture exam, that topic will not be included. A continuation of the key concepts, we just looked at comparing and contrasting the replication cycle of lytic and lysogenic bacteriophages and extruding phages. Now we're going to add to that with lytic and latent animal viruses. So you'll find some similarities here with animal viruses versus bacteriophages, right? So adsorption, penetration. In this case, there is one extra step here. So with animal viruses, the entire virion particle enters the cell. Therefore, there is one additional step, which is uncoating. This is the release of viral nucleic acid into the host cell. Once this occurs, same process as before in general, the host cell machinery takes over replication, transcription, translation to synthesize new virion proteins. These will then assemble, and this varies depending upon which virus we're talking about, and especially dependent upon naked versus budding or enveloped animal viruses. And then again, release. This would be either release through lysis or release through exocytosis or budding. This is from your book, just to show you these step-by-step processes. Again, here is a picture. You can tell in this one there's a nucleocapsid on the inside, and you can see the glycoprotein spikes on this circle around it. Therefore, you know we're talking about an enveloped virus in this picture here. Naked viruses also have those spikes, as you recall, but they're actually on that nucleocapsid. So step one is adsorption or attachment. In this case, it varies depending upon which cell to host, virus to host cell interaction, but there's always some type of host cell receptor that's on the cell membrane that is recognized by those glycoprotein spikes. Step two is penetration, so somehow that virus gets into altogether the cell, and then step three is uncoating. Usually the uncoating step is just the host cell machinery, the host cell digestive enzymes, breaking apart those capsular proteins in the envelope. We'll get to that in just a minute. And you have the release of a viral nucleic acid. Now at this stage, I will just let you know we won't go into any of the detail, but this viral nucleic acid must be processed in two different places since this is a eukaryotic cell. Replication occurs in the nucleus. Transcription translation occurs outside the nucleus, either in the cytoplasm or the rough endoplasmic reticulum. So every virus has evolved a different path at this point, depending upon what type of nucleic acid it has, and dependent upon what machinery it uses. So some viruses will actually take advantage of the various like post-translational modification machinery we have in our endoplasmic reticulum or our Golgi apparatus to produce modifications of their proteins. That's just an aside, just so you know that there are variations to this. But in general, the process, you can keep it as simple as this. So we have uncoating to release the nucleic acid. This nucleic acid is then synthesized or replicated, and then also proteins are synthesized through transcription translation using host cell machinery. In any case, there is a result of more nucleic acid, more viral proteins, and then we have assembly and release. And again, this varies depending upon if this is a naked virus or an envelope virus. So let's look at some of these in detail. So again, step one, adsorption or attachment. This varies dependent upon if this is, actually it doesn't really vary. So for the envelope viruses, the glycoprotein spikes are bound in the envelope itself. For naked virus, the virus glycoproteins, that's not true, but this is glycoproteins, so there's capsid spikes or glycoprotein spikes here and here. And they are different, but the general premise is the same. So let's go to our little chart here, same as we had before. So step one is attachment, adsorption. GP stands for glycoprotein. I'll just write that out first. Glycoprotein spikes are embedded in the nucleocapsid bind, oops, oh my goodness, bind host cell membrane receptors. Now the host cell membrane receptors, these are naturally occurring proteins that exist on the surface or the cell membrane of eukaryotic cells. Right now we're talking about animal viruses, so inside our body, our cells have receptors for cell-to-cell communication. Those are really important for how the cells normally function and the ability to sense and respond to things that are going on. So viruses take advantage of these cell membrane receptors, and we'll use them as their docking station. For envelope viruses, same thing. The glycoprotein spikes that are embedded in the envelope bind host cell membrane receptors. And this is, just as before, this is what provides host specificity, but not just host, it's host to cell specificity. For example, the new coronavirus that is going around China and is starting to cross borders. Scientists have just last week discovered the host cell membrane receptor that binds or that is recognized by the glycoprotein spikes on that coronavirus, and so are now able to at least understand and characterize that virus based on its receptor binding. And this particular host cell membrane receptor is located on only cells involved in our respiratory tract. Once that occurs, penetration and uncoating occurs. And this will vary depending upon if this is a naked virus or an enveloped virus. So here at the top we have a naked virus. This is just the nucleocapsid with those glycoprotein spikes. In this case, we have the binding of those spikes to a cell membrane receptor. This triggers the host cell to undergo a process called endocytosis or engulfment, wherein the entire host cell membrane folds in to create an internal vesicle. This is a process that happens all the time in eukaryotic cells and our cells, and the point of this is to take in large particles and then digest them. So this is a protected vessel inside the host cell, and the host sends machinery or enzymes to digest that stuff that's inside there. So this is the uncoating step here, is host cell machinery or host cell enzymes come and digest these proteins here, releasing that free nucleic acid. Here for the enveloped virus, remember, has a host cell membrane, the same cell membrane that it is binding to, essentially. So because of this, our cells have evolved to recognize this. This is actually a really common type of vesicle that's transported throughout the body. It's called an endosome. And so our cells already have naturally in place an ability to bring in these endosomes and not cause harm to the cell or to the stuff that's inside the endosome. And it does this by a process called membrane fusion. So here we have the binding of the glycoprotein spikes with host cell membrane receptors, but now we have something called membrane fusion, where essentially this envelope cell membrane fuses with the cell membrane of this bound cell. And so during this fusion process, you have the internal contents of that enveloped virus, which is the nucleocapsid, released to the inside of the cell. And then we have, again, host cell enzymes come and digest that capsid, the capsimer subunits, releasing that free nucleic acid. So let's go back and add this to our list. So 2, penetration. For naked animal viruses, this is a process called endocytosis, or the host cell membrane engulfs virion particle. Here for penetration, for an enveloped virus, we call this membrane fusion. We have the release of that whole nucleocapsid into the cell. For the naked virus, inside the endosome, there must be uncoating. So host, digestive enzymes break down protein, let's say capsumer proteins, releasing nucleic acid. Here for the uncoding step, we have also host digestive enzymes, same thing. It's just that for envelope virus, this is occurring in the cytoplasm. For naked virus, this is occurring inside these endosomes, these little internal vesicles, after the endocytosis process. Step 4, synthesis. So we have replication of viral nucleic acid, protein production, right? This is a highly regulated process. Transcription and translation are done using host machinery. I wrote some exceptions. Same rule applies as with bacteriophages. If the virus comes in with nucleic acid that is not recognized by our host cell, there must be enzymes that the virus brings with it to help change or convert that nucleic acid into a form that we can recognize and process. So here for step 4, this is going to be the same. Let's do the same as we saw before, replication of I'm sorry, I usually am a very good typer. Acid using host machinery. And the same process occurs over here. 5 is transcription and translation, synthesis, virus proteins. Same thing over here. Actually, let me add in one more. All right. Assembly occurs. This is a picture of a poliovirus infected cell just about to be released through lysis, right? The bursting open of the cell. It's actually kind of crazy because the host cell actually looks like these icosahedrally arranged capsumer subunits on the inside. So there is some rhyme or reason, some regulation to the assembly process. If this is an enveloped virus, then the viral spikes are put into the cell membrane prior to release. And I have a picture of that here, so I'll show you. Release is through either cell lysis, through a naked virus, right? So the bursting open of the cell. I cheated here. I put a picture of a bacteriophage lytic infection, but the same process occurs with animal viruses. Here is the process for an enveloped virus, which you'll see looks a whole lot like those extruding bacteriophages. So here we have the viral nucleic acid. After synthesis of viral proteins, those viral proteins are actually put right on the inside of the cell membrane. So here's this matrix protein. Here's the viral glycoprotein spikes that we talked about before. Once this process has been completed, then the viral nucleic acid, and in this case we actually have the capsumer subunits already surrounding this nucleic acid. So we actually have the nucleocapsid that comes and lines up right next to the cell membrane, and once this occurs it leads to exocytosis or budding. And now we have a new enveloped infectious virus that has a nucleocapsid on the inside, matrix proteins to help establish that cell membrane organization, and then the glycoprotein spikes embedded in the envelope. And this is an electron micrograph showing an active process of budding, where you have these nucleocapsids coming and being pulled or extruded out of that cell membrane, taking with it a part of that host cell. All right, so 6 would be spontaneous assembly here for naked animal viruses. That's not how you spell it. Spontaneous assembly, where you have the complete assembly and maturation virion particles inside the host cell. Here we have assembly of nucleocapsid in cytoplasm. And then we have the glycoprotein and matrix proteins embed or establish themselves at the host cell membrane. And then finally we have release. This is through lysis, so bursting open the cell. Usually there are late stage enzymes involved in actually creating that permeability in the cell membrane. And here we have budding release. Is through budding, which is when nucleocapsid lines up with that cell membrane at the site of this proteins and just pulls itself off. So a couple of these questions here. Why do animal viruses have envelopes and phages do not? Again, a good thought question. You can pause this and think about it. But I did mention this already. Just think about the host cell, not the virus, but the host cell. So for bacteriophages, their host cell is a bacteria, and bacteria all have a very rigid cell wall. And that cell wall, and especially for gram-negative cells, that lipopolysaccharide are very difficult to get through, essentially, for this process of exocytosis. So you would find it with animal viruses or with other eukaryotic cells that do not have that rigid cell wall, but any cell that does have the rigid cell wall would be very difficult to pass a cell membrane through, essentially, for that process of envelope budding. This next question, what are the similarities between the life cycles of, or replication cycles of lytic bacteriophages, and in this case, just think about naked animal viruses. This is one of those questions I've been asking. Just compare and contrast the replication cycle of a lytic bacteriophage and a lytic animal cycle, or animal virus cycle. And in this case, there's a lot of similarities, but there are distinct differences here, and you can go back to those charts that we just made to really understand those differences. So we do need to talk about duration. For animal viruses, we are talking about, much like we talked about before with bacteriophages, how they can be a productive infection or a latent infection. Same sort of concept occurs here, but it's just slightly different. So for animal viruses, we're talking about acute infections versus persistent infections. For acute infections, this one always has a short duration, beginning, middle, and end. Sometimes short is in quotations, so these can even last, like for mononucleosis, it is an acute infection, but it's a very long one, so you can even have that infection for about 6 months. But otherwise, we're talking about a few days to a few weeks for each of these viral infections. In all cases, your adaptive immune system kicks into gear and eliminates the infection, which also leads to memory response. So you have lasting immunity to these acute infections. Acute infections always are from productive infections, which again means virion particles produced at the end of a replication cycle for each infection of each cell. The infected cells will die, and actually this is also just varied. So obviously lytic viruses or naked viruses that lyse open the cells will kill the cell upon exit. Budding viruses keep the cell alive, but it compromises the cell membrane, so those cells will eventually die too, just not as rapid or not as quickly as the cells that are infected with lytic viruses. For multicellular organisms like humans, this process here causes direct damage. This could be localized to a particular region, or this could lead to widespread damage if this virus is something like a hemorrhagic fever virus like Ebola or Marburg, or any of these ones where the infected cells are actually throughout the entire body. Right, so this cell lysis can be very damaging. This also, of course, will lead to an immune response that also goes and lyses our own cells. So this process causes both direct damage, but also indirect damage. And in all cases, again, the host defense system will eliminate the virus. This is in contrast to persistent infections. These ones are long-lasting infectious processes, usually lasting the duration of the life of the individual who is infected. And there are 3 types that we'll write down here. But for this one, let's just kind of sketch out. I know it's going to be a little bit sketchy, haha, but let's just kind of write out what we're talking about here. And I need to get a better program to use, but for now I'll just write it. So for our acute, I don't know how to use this very well, acute For our acute stage, let's see. Okay. We have, oh shoot. Why doesn't it let me do it? Okay. Bear with me. Let's just delete this whole thing here. Yeah, okay. Let's start over. Okay. So here we have a really poor graph, an acute stage. We've seen this before. The progress of infection starts with this initial incubation stage, followed by that little prodromal period, followed by the peak of invasion, followed by some duration here where you're experiencing the worst signs and symptoms, and then eventually your immune system will kick into gear and eliminate this in that convalescent stage, followed by potentially a prolonged period of decline, right? So we remember each of these stages here from our chapter 11 on the progress of infection, where down on this axis is time, and over on this axis is either the signs and symptoms, or we could even write our extra axis. Remember we had this over here before. Let's change the color of this one if we can to, I don't know, turquoise. Okay. And say for this one, we can have the number of virus particles. We actually call it a virus titer. We'll see this again later on, but a titer means a count. T-I-T-E-R or R-E? It's E-R. Anyway, virus titer, so virus count. We're in the beginning, you are infected, and they do begin replicating, but this is log scale, so we don't really see it during that lag phase. Eventually, virus numbers start to go up, and as long as your immune system doesn't clear it right away, it just continues to go up. Eventually, your immune system will outdo or outnumber these viruses, and they will begin to go away. And usually they go away altogether. Sometimes it can take a little while for them to go away, right? So these follow that population growth curve. It's a little bit different because here, this is where your immune system kicks into gear, right? So your immune system outdoes the virus infection. At this point, remember we mentioned that if your immune system does not kick into gear, that virus count will just continue to go up. So it is possible that virus count can continue to escalate, and then this will always lead to the death of the individual, right? So this is acute, meaning there's a beginning, a middle, and an end. So let's write down acute for this one. A C U T E. So this is as opposed to persistent infections, which are long-lasting infections, of which there are 3 types. These are latent, chronic, and slow infections. So we'll go over each of these things. Now in this chart here, the x-axis is time. The y-axis can be signs and symptoms, or it can be virus titer, right? So how many viruses are released, or if they are releasing virus active virion particles. So the top one here, latent infection, the really classic examples of these types of viruses causing these infections are the herpes viruses, so varicella zoster, or the herpes zoster virus would be a really common one. This is the one that causes chickenpox and then later on causes shingles. This is also the same one that would cause continuous activation of virus in the form of like cold sores, or of course genital herpes, the same kind of thing. And in all of these cases, they're characterized by these periods of activation, where if you're just measuring this as signs and symptoms, you have a very classic increase in signs and symptoms that essentially peak, level off, and then you have convalescence, you get better. If you were to look at the virus expression, so how many viruses you are shedding or the active virion production, you would find that the virus numbers, the virus titer goes up initially. Your immune system kicks into gear and clears the infection. But in the case of herpes, even after your symptoms resolve, you have this non-infectious stage, but the virus doesn't ever clear. So in this case, the virus stays inside. In the case of chickenpox, the virus stays inside your nervous system cells and just stays in this non-infectious latency, right? And at some point, and sometimes we know the triggers and sometimes we don't, but there is a period of reactivation where the virus becomes productive again and you experience signs and symptoms and convalescence again, right? Again, for chickenpox, you would find it with the expression of chickenpox as a kid, usually, and then later on in life, in the 50s, 60s, or 70s, you would experience shingles or zoster. For cold sores, this would just be a continuous reactivation time after time. The second one here is a chronic infection. The hepatitis viruses, hepatitis B and C, would be really classic examples of this type of infection. These are not the only viruses that can fit under these categories, but this is really common. In this case, for hepatitis infection, if you were to look at signs and symptoms, you would have an increase in signs and symptoms, and then you stay infected and also symptomatic for the life of the human. But sometimes there can be this acute stage, and then you have no symptoms, but you're chronically infected. And so if you were to look at the virus count, the virus titer stays high for the entire life of the human. That's a chronic infection. So you would either be a carrier, an asymptomatic carrier, where you're not expressing symptoms, but you're releasing virus, or you could be a chronic carrier that you're symptomatic. So you have symptoms and you are also actively releasing virion particles. And the last one here is a slow infection. HIV progressing into AIDS would be the classic example of this one. And for HIV, you have again this initial period of acute stage. This is the beginning, middle, and end. It looks something like mononucleosis. The symptoms resolve. The virus count actually goes down to almost undetectable levels, but in the case of HIV, the virus invades a different type of cell. They get into your CD4 positive T cells, and after a prolonged period of time, begin to increase in numbers, and over time gets to a count where you become immunocompromised, and usually actually will always lead to death. Now, of course, for AIDS, we have medication that essentially halts viral replication. So this period of time can be completely prolonged and even prevented altogether from the development of AIDS. But if untreated, this is the classic look of these types of infections. So if we were to just add these things to my horrible drawing here, we can add our persistent infections over here, where we have 3 different types of persistent infections. In all cases, we have time on the x-axis and signs and symptoms on the y, and if we wanted to, we could also plot, just as before, we could do virus titers on this side. And in all cases, we have for, what are we doing here? This is latency. I need to get out my pen, sorry guys. For these periods of latency, we have the increase in signs and symptoms. They level off, you get better, and the symptoms go away, and then you might have it again. These short periods of increased signs and symptoms, etc. And if you were to look at the virus number, you would pretty much find the same thing, where the virus numbers rise, your immune system kicks into gear, the virus count drops, but even though they're undetectable, they don't ever go away. So you always have some virus inside. And but for these latent infections, really during that period of latency here, you really cannot detect virus. This is a non-transmissible stage, so virus counts are very, very low. Usually these things are undetectable in the blood because they hide away essentially inside particular types of cells in the body. For the chronic infections down here, these ones again would begin with an acute stage. You increase signs and symptoms, and either you resolve signs and symptoms or you don't. And these people are infected forever. So this would be signs and symptoms. Potentially you could have this kind of acute stage where signs and symptoms go away, or even you could be just asymptomatic forever. Right, so this varies depending upon which disease we're talking about, but for hepatitis, most of the time the individual is infected, the virus counts go up, and the virus counts stay at that consistent level, chronically shedding viruses for the entire duration of life, with actually no medication that could halt viral replication. There is some medication on the market now. These are antiretroviral therapies called interferon, which is a really cool process, and we'll talk about it a little bit more when we get into chapter 12 and 13 on the immune system. But for now, we'll just kind of leave it at this. And the last one is the slow. Again, you experience a period of acute or an acute stage, signs and symptoms increase, level off, you get better. If you were to look at the immune system, you have an active adaptive immune response kicking into gear, which readily clears that viral infection out of its primary infection, which is the first site. But then the virus moves on to a different region of the body, and over a prolonged period of time, right? And you can kind of draw this with 2 lines here to indicate that can be years, months, years, decades even, between this very, very first window and then the development of the full-blown AIDS for HIV infection. And in all cases, the virus count actually follows the same pattern, right? So for virus count, same thing, you would find the virus count goes up and actually goes down really, really low, but never goes away. And then over time, that virus count is increasing until the individual reaches the end of their life, so that's death. These are just slides showing you the same thing. This is the picture of a herpes infection causing cold sores in the mouth. Some people experience this. Once you have your first cold sore, then you'll have them for life. So if you've never experienced a cold sore, that means you are not infected with herpes. And you will not ever experience these until you gain that herpes infection. For chronic infections, again, hepatitis is the most classic example of this. The virus is ever present, so always expressing virus particles for productive infection. In this case, the symptoms are present or not, or they can come late term as the disease progresses. Like with the liver, you gain a different type of disease presentation, which is called cirrhosis, as the liver degrades. And then a slow, persistent infection. This is where you have integration of the viral genome. After that initial infection, you have that slow and gradual increase in the viral particles, no initial symptoms, except for that first acute stage. These are always lethal. This is a picture of Kaposi sarcoma, which is a really classic opportunistic infection in AIDS individuals and one of the classic AIDS-defining illnesses. So cytotoxic or cytopathic effects are, cyto means cell, and pathic is like the pathology or the damage or disease state. So what we were talking about with cytopathic or cytotoxicity effects is what the impact on a cellular level is for the virus infection. And these are very helpful for diagnosticians, so people who are taking these samples from patients and trying to determine what type of infection the patient has. Sometimes these cytopathic effects are enough to give a positive diagnosis without even doing any further testing on the type of virus. For example, these giant cells that are formed here are really classic of cytomegalovirus, I think is what we're looking at here. And these are inclusion bodies that are formed with another type of virus. So if you're trying to diagnose a patient and you get a blood smear, or depending upon which part of the body this came from, like a vaginal smear or something, then just looking under the microscope and determining the cytopathic effects might help alert the diagnostician as to what type of infection the person has. And then this last slide here, I think, is looking at the types of viruses that can cause cancer. We call these oncogenic viruses or oncogenic infections. This is cancer causing. These are always due to an integration of virus genome into the host genome. So we looked at this with, for example, in bacteriophages, the temperate phage can do this integration to create that prophage stage. Now there's something similar that happens with many of these animal viruses, the same concept of integration, but oftentimes it's not site-specific. And so the integration is random, and it can disrupt some really important or really pivotal genes involved in cellular regulation, which can lead to abnormal, all kinds of things. So it can lead to increased the rate of growth, or alters essentially the checkpoints in the growth cycle of a cell. Altered chromosomes, obviously, changes to cell surface molecules. Like when we get into chapter 13, we'll talk about this again with major histocompatibility complex, or MHC complexes. And then in all cases, we're talking about prevention of cell death. These are oncoviruses that cause these. Here's a few really classic examples. Human papillomavirus, or HPV, can cause cervical cancer, can even cause rectal cancer or penile cancer in men. Herpes and the Epstein-Barr virus, these are able to cause various types of cancers. Hepatitis can lead to liver cancer. This picture here, I just added it just to show some variations of what we just talked about. I don't really like it very much, so you don't have to use it if you don't want to, but just like we talked about with bacteriophages and the different types of infections that that a bacteriophage can cause, this is sort of showing you the same concept here, where we have a virion particle that infects the cell. So there's absorption, entry, and then from here we can have integration of viral DNA, where that integrated thing can become a tumor cell, like with Epstein-Barr virus or something along those lines, or HPV. Here we have integration, but it does not lead to alterations of the host cells, so this would be periods of latency, like with herpes. Here is where the virus can circularize and form a plasmid, and this plasmid has genes that are expressed that can also lead to tumor-causing alterations of that cell. Here is a lytic infection, something with a naked virus that we talked about. This is an acute infection where you have productive virion particles. Here are examples of persistent infections, in this case looking at a budding enveloped virus that can continuously leak out. This would be even something like HIV can do this. And here's another example, something that we saw over here, where we have integration of the viral nucleic acid, but this is in the form of a plasmid that can just independently replicate. But in this case, there is no expression of viral genes, and so you have latency established. This question is just a basic virus structure question. HIV attacks only specific types of human cells. Can you explain why a virus can enter some types of human cells but not others? Hopefully you're thinking about the glycoprotein spikes, and that's what provides specificity for virus with not just host species but also with host cell types. This one here, describe the adaptation of viruses that do not immediately kill the host and explain what its function might be. You should be thinking back to when we talked about filamentous or extruding bacteriophages, and we'll see the exact same question was asked then. What benefits does extruding provide a bacteriophage? Well, in this case, what benefit does budding, for example, or latency provide for a virus? And the same kinds of concepts you should be thinking about We talked about perhaps protection from the environment. In this case, now it's an animal virus, so this is definitely inside a human or another animal where we have a highly functioning immune system that's great at detecting foreign particles. So protection from the host immune system is a huge part of this benefit, or a huge part of this function. And then, as we mentioned before, you have continuous productive infection if this is a productive infection like a budding animal virus. So you can continuously have that, essentially that virus factory stay alive and make more virion particles instead of killing that cell altogether. And then of course we have the passage of that nucleic acid on to the host cell's daughters, right? So that would be another example of vertical transmission. How might mutations confer advantages to viruses? Now, this is especially relevant when we're talking about the annual flu that goes around and also HIV. These two viruses are really, really prone to mutations. And perhaps you have been taught that all mutations are bad. That's definitely not true. Mutations with the virus can confer some significant benefits in this regard. Mutations will lead to the host, which is, in this case, we're talking about humans. Our immune system does not recognize it if it's mutated. So we develop very specific responses to the glycoproteins, for example, to the hemagglutinin, or whatever the other proteins are that are on the surface of these cells. And when the virus mutates, that means that there's genetic changes that lead to different types of proteins being produced. Now our immune system doesn't have as good of a detection for that mutated protein, and so the virus can go essentially undetected and cause infection again. And we're going to stop here for now.